Hello, people. Hey, Mairead. Okay, can I see your faces real quick? I want to make sure there are human beings behind all these names. Alex McCarron shows up. Yay, Alex. Okay, people. Bimalak, where the hell are you? Are you in Plattsburgh or are you still over in Africa?
I'm still in Plattsburgh,
Okay, I'm sorry.
sadly.
That's okay. All right, people, guess what? As I promised last week, today is quiz day. I sent you each a quiz by email. I want you to go to your email, open up that quiz, and I'll give you about 15 minutes to do it, and then we will discuss. Okay? Can you check your email and find that? If you can't find it, let me know. I'll do a screen share of it. Actually, let's just do that. Although, go to your email so you can actually write the answers in, but I'll do a screen share so everyone can at least look at it if you're having intellectual difficulties. I don't even know what that means. So that's 8 of the 9 questions. Why don't you go ahead and do that whilst I shut the heck up? I'm going to mute. "15 minutes." So I brought up that ninth question now. That's probably the most complicated one. So take a look at that sucker now. I'm back. So can we talk about this? Can we talk about this, people? Can we talk about this? Okay. Name of the Xenopus embryo after the axes are laid down. So what's the process that causes the axes to be formed? That's gastrulation. The end of gastrulation is called a gastrula. So the answer we were looking for there was gastrula. Shape of the blastula in the zebrafish. It's kind of like a flat thing of cells, a flat pancake of cells. I'm not really sure exactly what I was looking for there, but basically it's not a ball of cells like the Xenopus. It's like flat bilayer, I think. Axis that the primitive streak goes along in the chick. That's from posterior to anterior on that AP axis. The first structure of what becomes the central nervous system. That is the neural plate. Oops, I got a chat here. Should we email you the quiz? No, grade it yourself and then tell me how many you got wrong, and I'll subtract that from your overall score in the class, okay? In other words, if you did well, congratulations. If you did poorly, let me know and I'll subtract it from your score. Okay, first structure of the central nervous system is the neural plate. Part of the Xenopus egg that becomes the endoderm, that's the vegetal pole. Mesoderm in the Xenopus embryo that becomes the body wall, that's the lateral plate mesoderm. Now, I think that's what I said. That's one way of saying it, but the other is the somatopleural mesoderm, somatopleural. Remember, there's the somatopleural and the splanchnopleural. And splanchnopleural becomes internal organs, and the somatopleural becomes the body, the body wall. Okay, three different germ layers. That's your ectoderm, endoderm, and mesoderm. Ectoderm is skin and nerve. Mesoderm is muscle, bone, blood, and endoderm is gut. Okay. What is the difference between neurulation in xenopus and zebrafish? In xenopus, during neurulation, the neural plate goes into neural folds, which then become a neural tube. In zebrafish, it does neural folds, but then it flattens into a neural keel, and then later opens up to make the neural tube. What germ layer does the notochord and somites come from? That's mesoderm. What tissues they become? They become the vertebrae, basically. During which stage do they appear? They appear during neurulation, during the neurula stage. Okay, again, I'm hoping you people knew all those answers to those questions. And again, if you did very poorly on that quiz, let me know and I will take points away from your overall score. If you did well, congratulations, you're smarty pants. Okay, so today's Monday. So today we will be doing mammalian, or in other words, the mouse development. And this is, no, never mind. After this, we're going to do a little human. And I think that's it for this chapter. We don't have an exam schedule until February 22nd. So we may end up doing some of chapter 4 or all of chapter 4 before that exam. So keep on your toes, people. Keep up with the material. Okay, so mouse. This is a mouse. The mouse starts as a fertilized egg, and these eggs are the smallest of all the models that we're talking about. And that's because they don't really need any yolk to grow. By the time they hit a certain stage, they're going to implant in the maternal uterine wall and get all of their nutrients from that. So these are very tiny eggs. And we will go from that egg to the blastocyst to the gastrula, or blastula, and then gastrula. And you're going to see with this mouse, we've got this cup shape, which is kind of weird. And then we will get from that a mouse. And these little suckers have got a really short life cycle. I mean, 7 weeks from fertilization to newly mating organisms. So imagine mice can double in number every 7 weeks. It's pretty quick, and they can really get out. And that's why cats are so important. And that's why the Egyptians loved the cats. Okay. So let's start out right after fertilization. So I did upload the notes for today's lecture about a half hour ago, maybe 45 minutes now. So they are there for you if you want to follow along. Okay, so the egg is released from the ovary, as we know, starts to come down the oviduct. As it comes down the oviduct, it's surrounded by a capsule of mucopolysaccharides and glycoprotein. So this is kind of like just like a jelly-like stuff on the outside. You can see it here in gray, and that's just kind of protecting that that egg as it's coming down the oviduct. And then provides a bit of a barrier there for the sperm to get through. But eventually, sperms being persistent, they will get through, and they will fertilize that egg. And once they fertilize the egg, similar to what we saw in, I believe it was the chick, the cells, the egg cell is frozen after the first meiotic division. So when the sperm fertilizes the egg, it goes through the second meiotic division, produces the final egg cell, and also another polar body. And then the nuclei fuse, and now you've got your zygote. So now you've got one cell, and the cell divides twice, to give you 4 cells, and divides again to give you 8 cells. Now at this point, you've got this bundle of cells together. We'll call that a morula, M-O-R-U-L-A, morula. And at this 8-cell stage, then it compacts, which means it just kind of squishes together. And when it does that, you can see how much extra morular space there is here, how much space is on the outside of this thing. When it compacts down together, there's a lot more cell-cell touching. And when we get into signal transduction, we'll see why that's important and the signals that are being passed then, and it changes cells into different types of cells. But at any rate, this is your morula, your compacted morula. Another thing that happens at this stage is the outside cells of the morula develop microvilli. So I think this picture down here is stole from the internet. I think it's, there's where I stole it from. I think it is intestinal epithelia, but it's the same kind of a thing. Your microvilli are little extensions of the plasma membrane with microfilaments inside of them, kind of arrays of microfilaments bound together to give it a little stiffness. So these microvilli then provide a lot more surface area for absorption of nutrients. So that's what they're doing. These outside cells then have microvilli on them. And then these cells will continue to divide until you get to this blastocyst stage. Now, this is where things get very interesting and very different from the other models that we talked about. On to slide 61. Okay, so here's the blastocyst stage. So you've got the hollow ball again, like we saw with Xenopus. Didn't see that with the chick or the zebrafish. And then we've got cells down here that are the actual embryo. So let's take a look at that back. This is not from your book. This is from a different thing. Here's a figure from your book. So here's your blastocyst. You've got two different types of cells here that all came from that original embryo. You've got these, actually more than two different types. So you've got these cells on the outside that are mural trophectoderm, and then you've got polar trophectoderm. So this whole embryo is surrounded by trophectoderm. That's going to be extra embryonic. It won't be part of the final embryo. And then here is the epiblast. That's what's actually going to be the embryo, or at least most of these cells will be embryo. And then here you've got primitive endoderm, which is not going to be part of the embryo. That'll also be extra embryonic. So you'll notice here, as compared to the other models, this embryo puts a lot of effort into extra embryonic tissues. And that's because that's where it's going to get its nutrients from the placenta in connection to the uterus. So a lot of the early cells then end up being extra embryonic. So basically you've got your inner mass of cells here, which is part of which will be the embryo. And then you've got this trophectoderm layer. And then the trophectoderm layer pumps fluid into the inner space here that forms that blastocele. Now this mural ectoderm is going to implant first when the fertilized embryo that makes it down the oviduct and hits the uterus. That's what's going to implant in that uterine wall. And remember, the uterine wall throughout the cycle, menstrual cycle, has been preparing for this implantation. So you've got proliferation of a whole lot of blood vessels and tissue there to get ready for this implantation. When the trophectoderm hits it, then it will implant. Okay, and as it does, so here's some vocabulary terms you need to know. Epiblast is the top part, and that is the actual embryo. Primitive endoderm here, mural trophectoderm, and polar trophectoderm. Okay, now we've gotten a little bit further along here. And we're in slide 63, is that correct? Slide 63, okay. So this is development of the blastocyst. So we're getting ready to get to the point where we can gastrulate. So what happens here is a whole lot of development of the extra embryonic tissues. At this point, these trophectoderm cells will divide without going through cytokinesis. So basically, you've got mitosis dividing the nucleus, making more and more nuclei, but without cytokinesis making separate cells. So you get these trophoblast giant cells, so big cells with a lot of nuclei in them, multinucleated cells. These are going to attach to that, attach to the uterine wall and release hormones that are going to cause the blood vessels of the mother to remodel, to change. So that's going to be the start of the production of this, what do you call it? Placental, what's the word? Oh God, I know I've got it here. Hang on a second. What's the word for maize? I don't know. I'll come across it. It is written down here later. But anyway, the placenta in general. Okay. So those the trophectoderm part becomes trophoblast giant cells, which implant into the uterus, remodel the uterus into something that'll be ready to become the placenta, basically. And lining the inside of that is this parietal endoderm, which I believe comes from the primitive endoderm from the last slide. And this is still covering the bottom side of the epiblast. And again, this is extra embryonic. And then the upper cells here that were, let's see, mural. Oh, well, the mural's the bottom. The see, this is the mural trophectoderm. The polar trophectoderm up here becomes this ectoplacental cone. So this will become the placenta eventually, and the umbilical cord. So you've got extra embryonic ectoderm here and ectoplacental cone here, and your epiblast down here. So the uterine wall then, once these trophoblast cells fuse with the uterine wall will grow around this whole thing, as well as it growing into the uterus. Okay. So now that you've got that primitive endoderm, it divides and becomes all this whole layer. And now it's kind of become two different types of endoderm now. This is the parietal endoderm, and this is the visceral endoderm. So all of that came from that primitive endoderm, and it's all going to be extra embryonic. Okay. So here's all your vocabulary words here, things you should know. Okay, late blastula. You notice you've got this cup shape to it, which is different from everything else we talked about so far, right? Especially if you go like zebrafish to chick, they were both like flat, and you had flat layers of cells, which kind of looks like it could be an embryo once it just kind of curves and now you've got your embryo. This is very different. It's got this cup shape to it. And the middle here is the amniotic fluid, basically. It's kind of called the proamniotic space. Epiblast, again, epithelial cells that are really the embryo proper. And then you've got your two different forms of endoderm. And at this point, you don't see the trophoblast because they've all become part of the placenta, and they've taken that part off of this figure, so you don't see it here. Now, let's see. At this point then, let's see, maybe the next slide. Okay, the next slide here. Ooh, primitive streak. Yikes. The primitive streak, that's just a bad joke, but I have procreated, I am a dad, and so I tell dad jokes. That's the way it goes. So anyway, at this point, you get kind of a buildup of cells here, and this is going to start the primitive streak. So because it's starting here, that will be your posterior end. And then the anterior end ends up over here, and the head ends up over here. So that's kind of your anterior posterior axis developing there. So now we're going to gastrulate. And the weird thing about the mouse in general, and this is specific to mice and not all mammals, it's inside out. And it needs to turn right side out later. So we're starting out inside out, which means your epiblast cells are here. They're going to divide, and then they're going to pass through the primitive streak to the outside of the embryo. So when you remember that the chick was like flat and they went kind of from the outside into the middle of it to form the 3 layers. Here we're doing it in the opposite direction. So these epiblast cells are going to divide and converge on this primitive streak, migrate through it, and then crawl across the outside. And similar to what we saw with the chick, they're going to go through the streak and go anterior and lateral to that streak. So that's going to fill out the next layer, the next 2 layers of the embryo, because before this, you've only got 1 layer. It's all epiblast. So this primitive streak then will advance downward in the anterior direction. This yellow out here is visceral endoderm, so that's extra embryonic. So as this passes down, your mesoderm is going to stay kind of central, while cells that become endodermal will go laterally and anteriorly. And those endodermal cells then will cover the whole epidermis, or the whole ectoderm it'll become now. So then your endodermal layer covers all of that. Let's back up a tad. And the dorsal side. And now another thing to note here, the amniotic fluid, you know, if you think about a normal embryo, like kind of hunched over holding its belly, amniotic fluid is on its back, right? Same thing here. Your amniotic fluid is on the dorsal side of the embryo. It's just that the dorsal side is on the inside, which again is specific to rodents and not to mammals in general, but we're inside out here. Let's see. So again, this is going to advance to the anterior side and then become a node when it hits that other side, kind of like Henson's node or the Spearman organizer. So it comes all the way down this way for gastrulation, and then here it becomes a node. Once it becomes a node, cells still go through that node and go forward. Remember, from the inside out through that node and going forward, and that's going to become your head process and precordial mesoderm. So that's very similar to what we saw in chick. I'm just looking at my notes here real quick. Okay, so once the node forms, that's the end of gastrulation. So the primitive streak starts here with cells from the inside dividing and going out, crawling across the outside and down along laterally as well as towards the anterior side. The primitive streak then progresses down to the bottom and it becomes a node. Cells continue to come out through that node, become mesoderm, and go forward again like that. And remember, just like the chick, these are ectodermal cells that once they go through the primitive streak, they become induced to become mesoderm and endoderm. So their cell type is changing as they're crawling through like that. Okay, what else must we say about this? Okay, so once you've gotten to the end of gastrulation, we're into neurulation. So this node is going to do the same thing as we saw in chick. It's going to reverse direction. So it comes down, forming the primitive streak, the node forms, and then it goes back up to neurulate. So here is just another view of it from your book. You've laid mesoderm down all along here. And if you can see, there's visceral endoderm on the outside of it. Okay, and then that visceral endoderm gets displaced by definitive endoderm, which is your new endoderm that will be part of the embryo. And that goes forward, as well as a lot of other mesoderm coming up this way, and that's making your head fold. And now you're ready to neurulate backwards. So now you can see this is your notochord is laid down by neurulation as that node went backwards up this way. So notochord and somites get laid down by neurulation. And just like we see in most other models, you get a neural plate forming on top of the notochord, and then a neural plate folds up to give you neural folds, and then it closes up to form a neural tube, closing in the middle first and then progressing anterior and posterior to close up that neural tube. And then everything anterior to the notochord becomes head here. And that visceral endoderm, if we back up a tad, so visceral endoderm is replacing, definitive endoderm is replacing the visceral endoderm, and then it will close up to form the gut, the foregut and the hindgut close first, and the whole thing closes up, and then you've got that. Okay, so that's kind of the progression from blastula or blastocyst all the way through the end of neurulation. Now, the weird thing about this rodent embryo, again, is like, here's your dorsal side. Really, you've got two dorsal sides hitting each other here, and ventrals all around the outside. So that's kind of weird. I kind of think of it like this. It's like as if a person was bent backwards. This is actually upside down. This is a little girl doing a back bend. So the dorsal side is here on the inside. The ventral side is all across the outside. The anterior is over here, and the posterior is over here. So what we need to do now is take this thing and flip it. So now the dorsal's on the outside and the ventral's on the inside, anterior here, posterior there. So it's kind of like going from a back bend to a forward bend. How is it going to do that? Well, the head and the feet come together and they kind of flip out somehow. And the actual mechanism kind of escapes me. I've seen it a couple of times. It doesn't make a lot of sense, but here we are. Here's your placenta that's forming from that ectoplacental cone. Head over here, posterior end over here, definitive endoderm on the outside where we don't want it. And so this then is going to bring the head and tail to come together. And then the tail moves past the head. And moving past the head. And then the whole thing flips. So what happened between the last slide and this one? It kind of escapes me visually. I can't really explain it very well because I'm not sure. What you really need to know is that it does happen. Okay, the head and tail come together and the thing flips. And now you've got dorsal on the outside, ventral on the inside. The ectoderm has divided and covered everything. So you've got skin all over everything. And at this point, you've got the brain forming. You're kind of far along here. You've already got an eye spot starting there. And here's your umbilical cord coming out of the ventral side. Okay. Do you have any questions about that? Vanessa, I see you talking. Are you talking to me? No. Thanks. Okay, no questions, comments? So again, the big thing that's different about this versus the chick is the chick was just like a flat disc that all the gastrulation, neurulation happened on. And this is a cup and it's coming from the inside out. And that's really the big change here. And you sure there are no questions or comments here? Discussion? Nothing? Because I'm kind of out of things to talk about today. Unless you've got, I do give relationship advice if you have any relationship questions. Not good advice, but advice. Nothing?
I had a quick question.
Okay.
So, you
Marry
talked
him.
about the
Marry
meso.
him. If he'll have you marry him. Oh, wait, that wasn't the question? Okay, I'm sorry. I'm sorry. Go ahead.
You said that the mesoderm forms the muscles, but
Yes.
a lot of like the digestive tract is also smooth muscle. Is that still endoderm or is that mesoderm?
Yeah, that's still endoderm. That's still endoderm. Sometimes, like with the chick, the chick's got a gizzard, and that's muscle on the outside, and that will be mesoderm. But mostly gut is all endoderm. Abdul Sabor, relationship advice? Do you have any for me?
No,
Yeah.
I think Yusuf needed some.
Okay. Yusuf Okine, where are you? I've got a bunch of chats here.
I'm straight. Don't worry.
Yusuf needs some. Steph too. Anyone have a Valentine? No, I've got a wife. But I got her a Valentine, so I'll still have a wife when that day's over. So life is good.
Doctor said you asked her to be a Valentine.
I'm sorry, what?
You asked her?
Do I ask her?
Did you ask her?
I think she just expects it.
Maybe it would like make her feel special. Like ask her, like, hey.
Mm.
Put a
I'll
little
ask her.
spark.
Ask her if she will be my Valentine, you mean?
Yes. It'll make
Okay.
her feel like
Well, there's some relationship advice that I will take. I'll do that. Will you be my Valentine? I'll ask her.
It's a
We
thing
shouldn't
everyone's
assume.
doing now.
Should never assume. Women don't like that. Okay. What else? Alex, you got anything for me? McKieran?
I got
What's that?
I got nothing. I don't know.
Nothing. Yeah. Yeah, I was like that when I was your age too, and I'm still like that, frankly. Okay, guys, let's call an end to this class, and I hope to see you all again here on Wednesday, and we'll talk about human development. Bye.